Good morning, everybody. Thanks so much for joining our webinar on wastewater this morning. I think I'm going to kick off straight away by asking Professor Himla Sudyal from the Academy of Science for South Africa to make some welcome remarks. Professor Sudyal, over to you, please.
Good morning, Angela. Good morning, colleagues, and thank you so much for joining us this morning on a very, very special webinar series. I'm standing in for Professor Musa Moshabela, who is the chair of the ASSAf Standing Committee on Health and Related Studies. Musa was very, very eager to be part of this session. He may join us a little bit later. Unfortunately, he has been called urgently to attend another meeting in KZN province. Colleagues, I want to start off with an insert that was published in Nature in May of this year by Heather Richardson, and I quote, wastewater testing can provide an unbiased snapshot of community health. Whatever access they have to healthcare systems, everyone has to go to the toilet. And as the effluent makes its way to treatment facilities, researchers can test it to determine what pathogens might be present. For more than 40 years, researchers have used water waste to monitor the spread of poliovirus, novovirus, influenza, hepatitis, and measles viruses can also be found in water. Unquote. And colleagues, today we are going to be treated by an exquisite panel of speakers spanning 3 different countries who are going to tell us how wastewater surveillance in the management of COVID-19 has taken place and share their experiences from these 3 countries. I mean, we have seen that data from wastewater testing is not meant to replace existing surveillance systems, but meant to complement them by providing an efficient pooled community sample, data for communities where timely COVID clinical testing is underutilized or unavailable, and data for the sub-country level. And so just hold on to your seats, because this is going to be an epic ride in learning how colleagues in the research area have been utilizing wastewater surveillance for the RNA SARS-CoV-2. So with that short preamble, because I do not want to steal the thunder from the speakers today, I now hand over back to the chair to introduce the proceedings for the day. Thank you all once again, and I'm so glad to see that we're also getting a growing number of attendees to this. So we all look forward to listening to you present to us how you have utilized this novel, well, not quite a novel, but this elegant technique to gain knowledge about the SARS-CoV-2 virus. Thank you, Angela, and over to you.
Thank you, Himla. That was a really great introduction to our proceedings today, and thank you so much also for standing in for Professor Moshabela. I absolutely agree with Himla that wastewater surveillance has really, again, in public health, proven its worth in helping us to manage this time the COVID-19 pandemic. And we have speakers from 3 different countries, diverse countries. And so obviously their experiences, the solutions they've applied to the task of rolling out a COVID surveillance system for SARS-CoV-2 has also been varied. So I'm really, really looking forward to hearing from all our speakers today about what the challenges that they have had, the solutions they've applied, and hearing about these successes in terms of mounting this wastewater surveillance-based system to help us manage COVID-19. So we have speakers from Nigeria, South Africa, and the UK. And the first one we're going to ask to present is Professor Vincent Chigo from the University of Nigeria. Professor Chigo, could you please share your screen and go ahead?
Thank you. Thank you, Professor Masi. Hello, everyone, and thank you for joining us on this webinar on wastewater surveillance in the management of COVID-19. I will be speaking on wastewater-based epidemiology for SARS-CoV-2 in Nigeria, the pursuits, prospect pursuits, and challenges. Okay, that's the structure of my talk. And about me, I got my PhD from the University of Forte in 2013, and back to University of Nigeria, where I am working now, and I lead the Water and Public Health Research Group. The Water and Public Health Research Group is actually a trans-faculty multidisciplinary group. So we have diverse research teams and areas of interest. We've had fundings and support and collaborators over the years, and we've also been productive. So, wastewater-based epidemiology for SARS-CoV-2 in Nigeria.
Just he hasn't seen it, but it comes up as
Christo, could you please mute your speaker?
Okay, so COVID-19 had been a huge global problem. And the counting as at yesterday, over 217 million cases had been reported worldwide, and with that, over 4.5 million deaths. And cases in Nigeria, it's approaching 200,000, but ranked based on confirmed cases, Nigeria, it's 19th in the world. And the current situation, as you can see, over about a little below 2.8 million has been tested. And it calls for concern because this just about 1.36% of the 201 population. So it's really challenging saying that there are fewer cases or not. We wouldn't know what it will be when more persons are tested. And but that's the situation now. Cases continue to be reported. It's and interestingly, the society is beginning, the population is beginning to carry on as if we've got used to COVID situation. The virus. The severe acute respiratory syndrome coronavirus 2 is the etiologic agent of COVID-19. That's the coronavirus disease that began in 2019. That's an enveloped virus. It's a positive sense single-stranded RNA with about 30 kb genome and up to 120 nanometer in diameter. Belongs to the family Coronaviridae. But while this was the immediate cause of the pandemic, mutations that resulted in the emergence of new variants. And a SARS-CoV-2 variant of concern is one that has demonstrated, an increase in transmissibility, a detrimental change in COVID-19 epidemiology, or an increase in virulence change in change in clinical disease production, the decrease in effectiveness of public health and social measures, available diagnostics, vaccines, and therapeutics. So whichever variant that meets any of these three is tagged SARS-CoV of concern. And so far we have the Alpha, which was first reported in the UK in September 2020. We also have the Beta variant, first reported in South Africa, still by December 2020. And the Gamma variant was first reported in Brazil in November 2020. And also the Delta variant that was first reported in India. But then we want to talk a little more about the viral structure. The virus contains 4 structural proteins: the spike, the spike protein, the envelope protein, the membrane protein, and the nucleocapsid. As you can see from the structure. And then it's got 16 non-structural proteins, out of which the NSP1 mediates RNA processing and replication, and NSP modulates the survival signaling pathway of host cell, and this is believed to separate the translated protein. Of course, with that nature of the virus, it can infect cells, and we want to discuss a little about how infection occurs and it's shared. It actually has an entry point into host cells in humans, and called the ACE2 receptor as the angiotensin converting enzyme 2. Now, why it's critical is that this ACE is expressed in the respiratory tract, the renal system, and the gastrointestinal tract. So SARS-CoV-2 can attach to and infect a wide range of human cells, the cells of our noses, the mouth, the lungs, and many other organs, and by so cause the disease. So what's the prospect for wastewater-based epidemiology? Looking into it, environmental surveillance by testing of wastewater for evidence of pathogen and monitoring disease has been of use in public health, as has been done in poliovirus surveillance. There is also surveillance going on globally for antimicrobial resistance. And of course, it's a major step in the One Health framework. The disease agents are excreted. It's known that disease agents are excreted in urine and feces of infected individuals, regardless of whether people are showing disease symptoms or not. And in the context of the ongoing COVID-19 pandemic, wastewater-based epidemiology is being used for the detection of SARS-CoV-2 shed into wastewater. And in WBE, the prevalence of SARS-CoV-2 infections in a community could be estimated by enumerating the virus RNA in the community sewage. But then changes in SARS-CoV RNA concentrations in wastewater sample collected from treatment plant influent have been shown to correlate with trends in reported cases. And that points to the prospect of wastewater-based epidemiology in COVID-19 management, which would include early warning of the occurrence of infection in populations, detection of asymptomatic infections within the population, detection of SARS-CoV-2 in locations where there is limited surveillance, and as well as in complementing clinical approaches. That's really key about the WB. It's complementary, it's not something that we want to say it will be used in isolation. Then, and if we achieve early detection and warning, then the result is that if we implement early stage interventions, critical pathways of exposure will be blocked and disease spread will be hindered. And the outcome will be that human lives are saved, and the social disruptions we've had will be minimized, and of course, reduction in the economic devastation that we have seen with COVID-19. But we had challenges in setting up the system here in Nigeria. And a key thing is what I'll call, number one is what I'll call the Nigeria Matters. Unique to us here, it might not be the issues everywhere. See, currently Nigeria has the highest number of people practicing open defecation. And only 14 out of the 774 local governments in Nigeria are open defecation free. This is a big problem, and storm events will only wash feces into water bodies. There is also the near absence of municipal wastewater. A population over 200 million. And yet we just have really about 4 municipal wastewater treatment plants. One in Abuja that was commissioned in 2007, and then 3 small and odd ones that are located in Nigeria's premier universities, one of which is where I work, the University of Nigeria. So with that, it becomes, why then are we talking of wastewater-based epidemiology here? How do we handle that? Another problem that is critical for Nigeria is that because we don't have municipal wastewater treatment plants, our cities depend largely on the septic tanks, which when they are filled, sewage is dumped into natural water bodies that are still, the society still depends on. Again, we have the problem of another big challenge, poor funding of research and lack of equipment. With this cannot be under, this cannot be, this is a serious matter, serious in the sense that research is poorly funded, and we don't have equipment to do things at the level that are done in other nations, and our institutions and universities and research centers are all struggling. Then we have also that even though we know that globally there are funders and grants, but institutional support for grant preparation and application is a big challenge here. Add to the Nigeria matter, there are also some technical issues. One is the reported low prevalence of COVID-19 in Nigerian communities. If that is so, then detecting the virus in waters will become another challenge. And then the issue is that if we are getting samples from environmental waters, there's a need for primary concentration. And as it is also prolonged storage of samples, and this should be at very low temperatures of -20 or -80. And this is challenging to maintain in the Nigerian environment where electricity, it's never steady or stable. Then also wastewater-based epidemiological SARS-CoV-2 is rapidly developing. So some protocols, sampling schemes, and now that we really don't have them, but as is advancing, the world and researchers are trying to do more. How are we dealing with the challenges? How should the challenges be dealt with? And there's need to expand beyond what we did here was really to expand beyond the collecting samples from wastewater treatment plants, also to include surface waters, since we know that they are always contaminated with physics in our environment. In our work, doing co-surveillance for other pathogenic viruses so that it won't be wasted effort. And we also engage in collaborative efforts that will maximize contribution. It's challenging, and we want to say really there is a serious desire in us here to go beyond, serving as sample collector. I want to become a real scientific contributor in collaborations that I have. But the limitations of lack of equipment, you find out that you get into international collaborations and you might not have equipment or the other to do what you want to do. While we prepared for this project, we did get a real-time PCR machine, but its level is below what I used in South Africa in 2010. That's some challenge. Also, I just also want to say that we can deal with these challenges if there is increased proactive engagement from institutions in South Africa and the North, Europe and America, with sub-Saharan African institutions. We are scientists here. We want to do well. We want to do all that needs to be done, and collaborations will help us do that. Having said that, what are we doing here in Nigeria? As the world struggles with the biggest public health crisis in recent times, what we did was to establish some collaborative relationship with the Abuja Environmental Protection Board. We are here in the eastern part of Nigeria, but for our interest in wastewater-based epidemiology, we had to link up with Abuja, and we have a working relationship with them. And we also did network locally and internationally. We moved on also to make 3 grant applications, one with international collaborators and one here for the National Research Fund that is supported by the Education Trust Fund. And well, at the end, only one was successful, and that's our UKGCRF Newton Fund Agile Response COVID-19 grant. Our project title is its co-surveillance of wastewater and environmental water samples for SARS-CoV-2 and other pathogenic viruses in South Africa and Nigeria. So we're interested in looking at the incidents and risk. And having secured that funding, we went ahead to recruit a postdoc into the project. But again, for funding and budget issues, not much has happened in the laboratory, but we're working a couple of review articles on virology about to be submitted, and a 3rd one had just been conceived. Again, there are 2 wastewater surveillance sample terms, the untreated wastewater at the influent, and then the primary source. But we are not dealing with the primary source here. We are collecting untreated wastewater and surface water samples from Enugu State, and in Abuja, the Federal Capital Secretary. We hope, because of the reported number of cases in Lagos, we hope, and we've established link to get samples also from Lagos, which is in western part of Nigeria. And with what we are doing, we also have a prospect of checking the impact of sudden demographic changes that are associated as schools resumes and students return to the campus. But so far, as it is, no sample analysis has been done to date. Then the question that comes to mind is, what effect would storage have on the results of our SARS-CoV-2 RNA detection? What effect would that have? Again, I've talked about the unsteady electricity in Nigeria. That will imply that even what we've kept, there will be freezing and thawing, and that could lead to marked decrease in the copy numbers of SARS-CoV-2 RNA. And a serious concern then is that the comparability of our studies with stored samples now with others in this project going on in South Africa and in the UK, we are beginning to seriously think that it will be low, But we are not giving up. We will contribute to global efforts, and wastewater-based epidemiology derived data on SARS-CoV-2 and other viruses. We mind is make up, and we have the belief, and we are doing all it takes. Well, we will find funding and collaborators, and aquatic virology will progress in Nigeria. Our project really is to be groundbreaking here in Nigeria, and that's part of why we struggled in setting up when budgetary issues from UCRI began to arise. But things are going on, and it's the interest is in wastewater-based epidemiology is increasing, and based on the people who had known that we are doing this out here, they got in touch, and I can confirm to you that the University of Ibadan is partnering with Rice University of the US to design and build an automatic wastewater sampler, confirming that WBE is a way to go. Of course, I won't stop. I will want to leave you with some co-reach, the rhyme of the ancient mariner that says, water, water everywhere, no any drop to drink. Why am I saying this? I want to say that water is critical, fit for drinking, or it's even become wastewater. So water-focused approaches are key for detecting viral outbreaks early in populations and in preventing spread to everywhere. COVID-19 took the whole world by surprise, and we must all work to contain that. So I want to say thank you very much. Thank you very much. And Nkosi Kaku. Dalo Norine. Thank you. And over to you, Angela.
Thank you very much, Professor Chigor. First of all, for an excellent introduction to the challenge of COVID-19 and telling us a lot more about the virus. And then also for just reminding us of the scale of the challenge in certain settings that hampers both the mounting an effective wastewater surveillance program, and also of the essential nature of sound systems. For example, as you mentioned, an electricity supply, a stable electricity supply. So I'm sure this raised many questions, but we'll leave it for the discussion section. And so I'm going to ask you to unshare your presentation right now, and then we will ask Dr. Renee Street from the South African Medical Research Council to start her presentation, which is titled Wastewater Surveillance in the Fight to Curb COVID-19 in South Africa. Renee, could you please start your presentation?
So, morning, everybody. Okay, Angie, can you see the screen?
Yes, we can, Renee. Please go ahead.
Super, thanks. So wastewater testing has been used successfully as a method for early detection of numerous diseases such as polio, norovirus, and hepatitis A. SARS-CoV-2 can be shed in feces of individuals who are both symptomatic and asymptomatic. So wastewater surveillance can be a leading indicator of change in COVID-19 burden in communities, as it represents a pooled community sample. It's a COVID-19 indicator that's independent of healthcare seeking behaviors and access to clinical testing. So wastewater surveillance went from being fairly marginalized to mainstream in a very short period of time during the pandemic. Currently, 55 countries are tracking SARS-CoV-2 in wastewater. This is happening across 7 continents. About 263 universities are tracking SARS-CoV-2 on their campuses. And from October, it'll be mandatory for wastewater testing for SARS-CoV-2 in all the European Union states. In March last year, the first researchers in the Netherlands isolated SARS-CoV-2 RNA from the wastewater site at the Schiphol airport in Amsterdam. And soon after that, many people realized that this could be used as a very important early warning system for COVID-19. The South African SAMRC scientists were following this key literature early on and wanted to explore the same early warning system in the South African setting. So in June, we started our laboratory proof of concept to isolate and quantify SARS-CoV-2 RNA from the wastewater. We were very excited the first time that we managed to do that. It happened in Cape Town, and soon afterwards we realized that there are numerous other things that needed to be discovered and explored. So we decided to take on a field proof of concept in Cape Town to test out logistics and a range of other interesting aspects. During the same time, we started thinking about extension to other sites, other parts of the country, and how to best start the early warning system. And just a couple of months later, in April of this year, the small project got escalated to the status of a program at the MRC. So it is now the Wastewater Surveillance and Research Program. The program was set up in a classic post-normal science situation where the facts are uncertain, stakes are high, decisions are urgent, but the program has moved forward with great agility and adaptability. The program consists of 5 units at the Medical Research Council. The Environment and Health Research Unit works on the coordination of the fieldwork and program facilitation. Our team, Biomedical Research and Innovation Platform, also known as BRIP, manage the laboratory work, and they are the reference lab for the program. We have support from my Biostatistics Research Unit on the data analysis. The Genomics Center, the MRC, works on the identification of the SARS-CoV-2 strains, and our TB platform also provides support in sample collection, data extraction, and analysis. So as our program was getting started and developing, we were exploring and debating key issues around wastewater sampling and the availability of the metadata. So there are two types of wastewater collection. There's a grab sample collection or composite sampling. Grab sampling is taking a scoop of wastewater from one location at one time point, whereas composite sampling takes multiple grab samples over a period of time and usually about in a 24-hour period. But the composite samplers themselves are kind of like mini bar fridges. They are very costly. They cost anything from 60,000 Rand to about 250,000 Rand. And generally at our wastewater treatment plants in South Africa, they are not available. Some of our plants have them, but it's not the norm. So to set one up is expensive. They also need some level of technical expertise. Of course, they also need electricity. And as Vincent mentioned, similar to in Nigeria, in South Africa, we have power cuts, power outages, also known as load shedding, for a couple hours at a time, which can definitely affect the composite sampling. And of course, it's a high-risk asset that can disappear. So there is that risk. So we realized quite early on that grab sampling, the once-off at one time point, was the best way to go in our context. Another key aspect that we were grappling with was the metadata we would need. So one way of normalizing or standardizing data so that one can compare results across wastewater treatment plants, we would need data like the daily wastewater inflow data, so what's coming into the wastewater treatment plant, as well as the population data. Well, this is possible for some sites. Generally, in our context, this data is just not available. Even more so when designing an early warning system and realizing that that data needs to be available within hours of sample collection. So if we couldn't account for flow and population, we needed to see if at least the trends between the non-normalized and the normalized data were similar. So in early July of last year, we set out to answer some of our key questions and started a field proof of concept study. We sampled from all of the 24 wastewater treatment plants in Cape Town over a 6-week period. And we were quite, our results were in line with the case data. You can see here was our first map of all of the wastewater treatment plants and their catchment areas on the 6th of July. And the red indicates high SARS-CoV-2 viral load in the wastewater, and the green moving from mustard yellow to green. And you could see going from left to right, there was a lot less red, smatterings of red here, and moving towards the 3rd of August, a lot more green. And this was definitely in line with the case numbers, as you can see here on the left, which were coming down nicely during that period. So we definitely had a lot of more confidence in the data over this period, matching it up with the COVID-19 case numbers. And during that time, we also managed to get where we could a couple of composite samples and saw that we did have a good correlation with our grab samples. But those composite samplers were also affected during that period by, as we've said, load shedding and various other issues. So it definitely did make us realize that grab was the only way to go for us. Just to share some of the data, we managed to do daily sampling at 8 wastewater treatment plants in Cape Town as well. And the city of Cape Town gave us the flow data and the population data for the select wastewater treatment plants, which we did the daily variation trends. On the Y1 axis on the left, you can see the non-normalized log transformed data, which was is in SARS-CoV-2 copies per mil. And on the right, here you can see the normalized data, which after adding and adjusting for the flow and the population, it's in SARS-CoV-2 copies per day per 1,000 inhabitants. And what we were looking to see, and we were happy to see, was the trend was very similar between the blue non-normalized data and then the red normalized data. So yeah, this was just stacked graphs showing a similar trend, and the correlation between them was very good. So although we know that the gold standard is to provide normalized data, we were content with these findings, and it supported our decision not to provide absolute data in SARS-CoV-2 copies per mil, but rather to categorize our findings going forward. So our program developed with 6 key elements. We decided moving out of Cape Town to work not only in the metropoli, but also small, medium towns and special communities, as well as unsewered areas. One of our key elements was a research aspect, working on genomics and variants, vaccine efficacy, and a range of other emerging research questions. Our wastewater sampling grew to a strategic selection and a growing network of sites, which we realized as well that it was important to standardize that sample collection, which we developed then a sample collection guide, that everybody was doing a similar type of sample collection across all of our sites as we developed. The laboratory analysis was important to standardize and make sure we had all the quality controls aspects in place. So BRIP in Cape Town is our reference lab for all of the analyses that we do. As we were onboarding various universities and partner universities, capacity development is a key aspect of this program. So we work also with under-resourced institutions and supporting staff and students in learning about the methodology and also providing ongoing support as the program goes on. We wanted to do this as an early warning system, so to get the data available to as many people as possible as early on. So we have developed a public-facing dashboard and also provide stakeholder communication on a weekly basis. Our research also has a lot of reach in terms of the science and public engagement, where we have lay articles, social media, and numerous media statements. Just to share, this was the City of Cape Town, back to the City of Cape Town from early November, where we started collecting wastewater from all 24 wastewater treatment plants again. And this was the start of the second COVID-19 wave in South Africa. So in this graph, and because it fell over December-January period, our results per wastewater treatment plants were definitely affected by the movement and the summer holidays and people being a lot more mobile. So in this graph, we took an average of all the wastewater treatment plants, and so it represents one SARS-CoV-2 RNA signal average per week. But you can clearly see the trend that as the SARS-CoV-2 wastewater signals going up in blue, a week or two later, the cases came up. And as our signal is coming down, in Cape Town, the cases came down a week or two later. So this was done using the lowest smoothing. But the highest number we ever saw with this average was around 3,000 copies per mil. But just fast forwarding a tiny bit, this is what we're now seeing on that same data line, but just adding a couple of weeks. So this is, of course, in South Africa, what we're now calling wave 3, where the Delta variant was introduced. So what we saw here with the 3,000 SARS-CoV-2 copies per mil as being one of our highest, the Delta variant has now knocked that out of the park, and it is so much, our average is so much higher. So in the beginning, we had early debates around, obviously the signal was so much higher. So is it that more people have COVID-19 but aren't testing? Of course, the testing algorithms affect the case data. Thought maybe more people were asymptomatic, but as the data became available around the world, we were seeing that, of course, the Delta variant people are shedding more of the virus. So what we were discussing and describing in detail before for our wave 2 with our data team and talking about outliers, everything really did shift for now our wave 3 in terms of the outliers became the new normal. Sorry. So currently we are sampling over 70 wastewater treatment plants across 4 provinces in South Africa. We sample from Limpopo province in Mopani and Vembe districts through the partnership with the University of Venda. In Gauteng, we sample in Tshwane Metro with the partnership of Sefago Magato University. In the Eastern Cape, we work with 2 metros and 1 district with the University of Fort Hare and Nelson Mandela University. And in Cape Town, we work in the city of Cape Town and 2 smaller districts, Breda Valley and Theewaterskloof. We will soon be adding 2 additional provinces, the Free State as well as KwaZulu-Natal, to our partner provinces. So our program is run, I do joke about it's run like a military operation. Every Monday morning, sampling teams that we have trained go out to the various wastewater treatment plants, to the sampling sites. And by midday, the samples need to be delivered to the laboratory because the samples are handed over to the lab scientists who then extract the SARS-CoV-2 RNA. The extraction process takes a couple of hours. So we don't want them to be working too late into the night. So we try and get the samples to them as soon as we can. On Tuesday, the PCR analysis happens, as well as the SARS-CoV-2 RNA quantification. And on Wednesday, the 4 partner universities, together with the reference lab at the MRC, have a lab huddle and discuss their data, any issues they've had, any challenges, and they get the QC's sign off to then send to our data team. Our data team does the statistical analysis, preparation of graphs and maps, provides information, collates information to key stakeholders and provides updates on our MRC dashboard, which then gives the health stakeholders 2 days, at least the Thursday and the Friday, to have any interventions required at facility and community levels. This is an example of our public-facing dashboard, which is updated weekly. It can be categorized or filtered, by the provinces we're working in, by the metros or districts, by the name of the wastewater treatment plant, and by the data categories. So previously I mentioned that we didn't provide absolute data. We are now providing data in categories. So the lowest category, level 1, is so it's 0 to 500 copies per mil of SARS-CoV-2, going right through to our highest category, category 5, which is over 7,500 copies per mil. And this is how the data is then presented and shared. We also have social media posts going out in our local languages to drive traffic to the dashboard and to alert people about how they can find out more about this work that we're doing and the trends in the area. Here's an example of a stakeholder update. This was from last week. This went to Mopani district in Limpopo province. This at a glance slide, you can see this this bar on the left is last week's, you can see the category data for the previous week. The one on the right is the current week, and then a trend indicator to see if the trend went down, it plateaued, or increased compared to the previous week. We also provide orientation maps of where the wastewater treatment plants are. And this is, of course, the historical trend from where we first started sampling in those wastewater treatment plants to date. Just another example of one of the metros, Nelson Mandela Bay. Again, historical trends are provided, and the weekly updates compared to the previous week, as well as the orientation maps. We also grouped the wastewater treatment plants that we're working on by province to see how many are in the low category, category 1, and how many are in the high category. This again was from last week, and you can see that the Western Cape has a number of wastewater treatment plants in the high category, which aligns with the case data. A couple of weeks ago, this looked very different, where Gauteng province was the epicenter. There were so many COVID-19 cases in that third wave in Gauteng, and there were a lot of wastewater treatment plants in the extremely high category 5. So this work has brought us together with so many interesting and dynamic partners, because some of them are local, regional, and international. What we found so interesting in this program is that in the beginning, we were reaching out to people, asking for their advice, and we got so many kind and generous responses of people just willing and able to help us and offer support of whatever we needed with technical expertise. And now we're finding that people are actually reaching out to us in our program asking, how did you do that? Can we learn from you? Can you assist us with various aspects? So we're really keen and excited to be able to share all this knowledge that we learned over the last couple of months with others who are also moving forward with this type of work. This work has attracted so much local and international media for the program, which has been really nice and dynamic. And yeah, we've had so many different diverse groups reaching out, asking for various interviews and podcasts. So that's been really rewarding as well. So a couple of lessons learned as I wrap up. One is that definitely with wave 2, we saw that our results were affected by the seasonal events or holidays. There's high variability in SARS-CoV-2 RNA in some of our wastewater treatment plants over some periods. Some it's hard to see a trend. I guess it speaks to the mobility of the urban population. We do need more time points, and actually multiple years of data is required to infer seasonal trends, but we are getting there. The case data is often not available by the wastewater treatment plant. So the wastewater treatment plant catchment area is difficult to how the health teams describe their health boundaries. So it's difficult to overlay where the wastewater treatment plant catchment are and to match together with the different cases. That can be quite tricky. And of course, everyone thinks it's the gold standard to match the case data, and you're seeing the trends. And although it's exciting when you do that, and we have done that in some cases, the case data is also affected by, it only affects the diagnosed cases. So it's affected by testing algorithm changes when, sometimes some areas say, please, nobody above the age of or below the age of 40 can get a COVID-19 test, for example, because the system is overburdened. The reporting of the rapid versus the PCR tests. And of course, just an example, in Durban recently, there was the civil unrest, which did affect people coming forward for testing. So there are many different things that can affect the cases that we're trying to link up our SARS-CoV-2 data with. And of course, as I demonstrated briefly, that each wave has behaved so differently, and we've learned so much from each wave, but we are continuing to learn. So our way forward is the expansion of our network, further training and capacity development for our university partners, working in defined communities, retrospective data analysis, which is really interesting, looking at the variants of concern, which my colleagues Rabia and Prit will touch on after this presentation. Which of course, the overall aim is to reduce morbidity and save lives. We have set up this fantastic SARS-CoV-2 RNA research platform, but we now intend to, in discussions with numerous people, to look at, for example, chronic diseases, pharmaceutical, illicit substances, and micropollutants as a platform for public health research. And lastly, just with thanks and appreciation for so many people that make this program a success, our academic partners, funders, communities, scientists, our think tank, and the municipalities, and just a big thanks to all the MRC investigators as well. Thanks very much.
Thank you very much, Renee, for telling the story of wastewater surveillance in South Africa, and for reminding us again of the challenges that we experience. And just like Nigeria, South Africa is hampered by electricity interruptions. The civil unrest of last month also had a huge impact on our systems, and for also showing us how you're responding to some of the challenges, doing the extra research, for example, showing that the collection of grab samples is robust, in the face of the challenges we have around continuous sampling. So we're going to move on to our next presentation, which is a presentation in two parts by Professor Rabia Johnson and Pratika. Before I hand over to them, I'd just like to welcome all the additional participants in this webinar and invite everyone to add to the chat and to the Q&A facility. In the discussion period afterwards, we'll try and respond to as many questions as possible, but of course you can always contact people individually. They have been sharing their email addresses in their presentations. But right now, I think Professor Rabia Johnson is going to be our first speaker, and she and Pratika will be focusing a bit more on the variants of concern and how that's been occurring. And I think she's going to use our work in Cape Town as an example of that. So Rabia, thank you very much. We'll hand over to you first. Rabia, unmute yourself.
Oh, thank you very much, Angie, and thank you very much for the kind introduction. I'll be sharing my screen quickly. Angie, are you able to see my screen?
We're not seeing it yet, Rabia.
Okay. Can you see it now?
There we go. There we go. Yes, we can see it. Please go ahead.
Okay. First and foremost, I would like to say thank you very much for ASSAf for allowing us this platform to showcase SAMRC's work on wastewater-based epidemiology. And then I would also like to say good morning, good evening, and good afternoon to all those dialed in on this webinar. My name is Rabia Johnson, like Angie said, and I will be presenting SAMRC's how SAMRC use wastewater-based epidemiology to track and trace key mutations linked to variants of concern in 17 wastewater treatment plants in Cape Town, South Africa. This will then be followed by my colleague, Dr. Pratika Ramarak, who will provide structural insight on the mutations and how this links to the variants of concern and their role in cellular infectivity. So with no further ado, Renee gave a very nice introduction to wastewater-based epidemiology, and through the course of the pandemic, this has been used globally as well as at SAMRC to detect and to quantify viral load within wastewater. More recently, we started with monitoring defined communities as well, and I will give you one slide and an overview of how we monitor defined communities such as your university residences. And this is quite important because, you know, there's a lot of movement within university residences, and if you see an increase or a spike in viral load, you need prompt response and informed decision making. And then in April of 2021, we started with the tracking and tracing of key mutations linked to variants of concern. And why variants of concern? Because this is important for because mutations linked to variants of concern increase transmissibility, it also increase disease severity, and it decrease it reduce your vaccine efficacy. And for those reasons, we need to monitor and screen for variants of concern, especially currently with our vaccine rollout. So this graph is one of our graphs where we looked at the viral load or detecting SARS-CoV-2 viral RNA within university residences. If you look at the bar graphs represents the daily case data. Like my colleague said, South Africa is currently experiencing its third wave. This is data from one of the local universities in Cape Town, South Africa. And here in Cape Town, we experienced our first wave round about in July, early June. And here you could see the line graph that overlay the bar graphs, which is the case data. The line graph represents the viral load that we were screening within those university residences. Taking a closer look at that, here you can clearly see on your y-axis is the genome copies per mole. And this is the start of the third wave in the Western Cape. And we were screening at the time 5 university residences. And quite interestingly, we could see there was an increase in the viral load at the start of the third wave. And then this data was then taken to university management. They then decided that classes must go online to restrict movement, and some students went home. And here immediately we could see that with that, the restrictions put into place, we could see there was a decrease in the viral load. That was then followed by the recess or break, winter break, and then universities opened again in late July, early August, and once again, because classes is not online anymore, students are back at the university, and we could see this increase in the viral load. So next, what we will be doing is retrospectively screen those samples to see what was the circulating variants within those university residences. And speak about variants. Through the course of the pandemic, several rapidly spreading variants of concern have arisen in both the United Kingdom, South Africa, Brazil, and India. And we came to know of it by the name of the Alpha, the Beta, the Gamma, and the Delta variants. And these identify lineages are termed variants of concern, and it depends on their propensity to infect and transmit due to the multiple mutations that is found within the spike protein of these genes. And then one can ask ourselves the question, why surveillance? Surveillance is important because you need to understand when a sample was introduced, what sample is currently circulating within that area, and also how that sample, or not sample, or how that variant, when the variant was introduced, what is currently circulating, and what's the geographical distribution of that circulating variant within that catchment area, because that would then require for prompt response and informed decision-making by our local authorities. So before I showcase you our data, I just want to show you how the clinical data and the data that has been sequenced in South Africa coincided with the wastewater data, and how this is NGS sequencing. And I will then present our data, which is data that we use quantitative real-time PCR to detect mutations linked to variants of concern. So this graph depicts the NGS sequencing data from clinical data. This is the latest data taken from their website, and it was up to the end of August. So in South Africa, the second wave started in December and it ended in late January. Then if you look at, and at the time, the beta variant was the most predominant variant. If you look at the graph of the NGS sequencing of the clinical data, you could clearly see that the beta variant dominated in all provinces in May. However, following its detection in week 18, the Delta variant became the dominant variant. And here you can nicely see the pie graphs, June, July, and up to now, August, you could see these were the dominant variants. Because I'm presenting data, quantitative real-time data for the Western Cape, I also would like to give you an overview of the NGS data for the Western Cape. Once again, I'm showing you on the y-axis the number of cases, and then on the y-axis on the left-hand side, the percent of genomes that has been sequenced. If you look at the black line graph, that is the case, the case data, the clinical case data, and then the bar graphs represents the different variants. So here, NGS data show that in the Western Cape, once again, if you look at Epi week 1, you could clearly see that was the end of the second wave. We could see that the beta variant was dominating. And then when we move to Epi week 21, you could clearly see, now we see there was a shift with the Delta variant currently being the dominant variant within the Western Cape. So one can ask yourself the question, so why would you go for quantitative real-time PCR? Why not using NGS sequencing? Because at the end, NGS sequencing is your gold standard. So most studies use next generation sequencing to gather information about the diversity of SARS-CoV-2 variants at the community level. Now, NGS sequencing is informative to identify new variants, but it is also costly to perform routine screening of switch samples to identify and profile known mutations associated with these variants of concern. Furthermore, one can also argue that in most low and middle income countries, they don't have access to NGS sequencing, but they do have access to a PCR machine. So it will then be a cheaper and more robust method to use. Also, NGS sequencing, though it is the gold standard and you can identify novel or new variants, for routine surveillance and to get a result within 24 hours and so that you can make an informed decision and to understand what is currently circulating, the PCR-based method would also then be a cheaper, more robust method to use to perform this routine surveillance. Now, bringing it back to wastewater compared to clinical samples, if you're looking at sequencing of one genome of a person, if you take a swab sample, that will be representative of one strain that dominates within that person. It will give you an understanding of what is happening within that person. However, sequencing or even doing quantitative real-time PCR or genotyping PCR using wastewater-based epidemiology, because wastewater-based epidemiology investigate nucleic fragments shed from multiple infected individuals. So this will give you an overview of what is currently happening or what is currently circulating on a population level, and not just one, give you an indication of what is happening within one person. It will actually tell you in that catchment area, what is the circulating strain within that specific catchment area. So genotyping of these fragments can identify circulating variants and its geographical distribution within a given catchment area with much less effort than your clinical surveillance. And this is also especially important with the current lineage of SARS-CoV-2 that is constantly mutating, affecting vaccine efficacy and viral transmission. And understanding the dynamics of this highly virulent strain will allow will allow your health officials for prompt response and informed decision making. And that's why we opted for a catchment assay. So even though our samples is currently being submitted for whole genome sequencing, and SAMRC do have a sequencing platform where we do sequence our samples, we also perform routine screening, and with routine screening we are able to give a result within 24 hours, and we make use of a catchment genotyping assay to screen only key mutations linked to the variants of concern. And this then allows to identify hotspots while tracking temporal trends of the variants of concern. And this will also give us an indication of the prevalence and frequency of these mutations that we can then measure over time. And for this, we investigated since April 17 wastewater treatment plants, and we screened approximately 272 samples. So this is just one of a figure depicting the mutation frequency that we are currently screening for, and this are the variants of SARS-CoV-2 found in the sewage over a 3-month period. So for the mutation frequency, we screen key mutations for the beta, the alpha, as well as the delta variant, and you can add more mutations to this. So for this, I'm just showcasing some of the mutations that we are currently screening for. And here we could clearly see, if you look at the frequency of key mutations for the beta variant, which is your E, your K417N, and the 501, you could clearly see that prior to the onset of the third wave, we could see in these week 17 up to approximately 23, you could see that there was a high frequency, higher than 60%, for the specified mutation that is linked or that is key to the beta variant. Then, if you look at mutations key to the delta variant, it was quite interesting to note that in these wastewater treatment plants, these mutations were picked up as wild type, and then from week 20 onwards, if you look at 20 onwards, we could see that the frequency of these mutations increase, while those of the beta decrease, where we found that the frequency for the the frequency for the delta mutations increased to above 90% throughout. Then we also look at some mutations, and these are the 681H is one of the mutations for the alpha variant. What was quite interesting, we saw this erratic response of this P681 mutations, and all of a sudden from week 21 onwards, the frequency of this P681H mutation that is linked to the alpha variant, we could also see that this specific mutation, that there was an increase in the mutation frequency, also above 80% throughout. So this is just a temporal analysis of the variants and the distribution over time using our quantitative real-time PCR. So again, this is just a quick overview. I want to bring your attention to the line graph, which is our current diagnosed COVID-19 positive clinical case data, showing from week 21 onwards, we could see that's the start of the third wave. There was an increase in the viral, in the case numbers. With that increase, we also saw an increase in the viral load within wastewater treatment plants. But the dynamics change a bit. With that increase in the viral load, we could also observe that there was a decrease in the beta variant, while the delta variant, which is the green bar here, you could see became the prominent, and there was an increase in this the delta variant from the onset of the third wave. Thus, our data suggests this heightened prevalence observed during the third wave could have been possibly driven by the delta variant here. This is just a closer look at the temporal analysis. and to identify if the Delta variant was definitely the prominent variant during the third wave. Here we could look at the Delta variant distribution. Again, we could see we couldn't detect it, and then from week 20 onwards, the Delta variant became the predominant variant. If you look at the beta variant, you could see there was a decrease in the beta variant. However, one mutation that was standing out, and this mutation is linked to the current alpha variant. What was quite interesting was the fact that while the Delta variant became the predominant variant by the end of June, and this confirmed the NIC data, we saw a co-occurrence of key Delta mutations with a 681H mutation. If you look at the gray bar graphs here, we could see that there was an increase in the 681H mutation that co-occurred with the Delta variant. So then we ask ourselves the question, so when was this Delta variant then introduced into our wastewater treatment plants, and which wastewater treatment plant was detected in first? With our spatial and temporal mapping, we were able to answer that specific question. Once again, if you look at prior to week 20, you could see that the beta variant was the predominant variant. And if you ask yourself the question, when was it introduced? When we had a closer look, we could clearly see spatial mapping showed that the Delta variant was introduced in the Gordons Bay as well as the Melkbos Strand area, where after it then spread to the remaining catchment areas. And here you can see the latest graph where you could see that most of our catchment areas now has, we are able to detect the Delta variant within these different wastewater treatment plants. And collectively, it suggests that the wastewater-based epidemiology, based on our results, we conclude, can then be used as a proxy for prompt and informed decision-making. So now I will hand over to my colleague, Dr. Prit Ramarak, and she will give you a quick update and explanation of what these mutations within these variants of concern means for cellular infectivity. Thank you.
Thank you, Prof. Johnson, and good morning, ladies and gentlemen. It's been absolutely fascinating to observe how the SARS-CoV-2 has evolved on a molecular level in an attempt to increase infectivity and overcome prophylactic therapies. So based on this rapid spread of the Delta variant that was assessed in the wastewater surveillance, we took a different approach to evaluate the potential structural mechanisms by which these variants have led to increased infectivity. And we did this using in silico molecular modeling techniques, including molecular docking and interaction analysis. So firstly, just a brief overview of where the characteristic mutations are seen in the variants of concern. So the alpha variant of concern is characterized by a 2 residue deletion that is seen at the N-terminal domain, a mutation at the S1, S2 boundary site, as well as a mutation at the receptor binding domain. And this is seen in green. The beta mutations are all located at the receptor binding domain and are seen in red. And the delta mutations are located at the receptor binding domain and S1, S2 boundary site and are seen in orange. So during viral infectivity, what occurs is that the receptor binding domain of the trimeric structure, which you see on the left, confers or modifies into an open conformation. And this initiates binding of angiotensin converting enzyme 2, also known as ACE2. So once ACE2 binds to the receptor binding domain, as you can see on the right, it shows where the receptor binding domain is. So once this binds, the S1, S2 boundary site is cleaved by a protein known as furin. And once that occurs, you have structural modifications occurring at the HR1 and HR2 peptides, which then allow for a structural modification of the spike protein. And that allows for attachment and fusion of the viral particle to the host cell membrane. Once this fusion occurs, the viral RNA is then released into the host cell for further replication. So based on where these mutations are located on the spike protein, we assessed two of these main binding regions, which is the receptor binding domain seen in dark in medium blue, as well as the S1, S2 boundary site seen in purple. And this was done by comparing the wild type versus the mutants when bound to ACE2 and furin respectively. So next slide, please. So to identify and compare the binding landscape between the viral and the host proteins, the mutations and deletions were computed as per variant of concern. And this was then followed by molecular docking and interaction analysis. The mutations that were observed at the receptor binding domain, as you can see in blue, have led to clear conformational shifts of the way that the ACE2 binds to the receptor binding domain. And this is seen by an increase in surface area interaction between the two proteins. And it was clear by our analysis that the beta variant displayed the strongest binding affinity to ACE2, which makes sense because most of the mutations seen in the beta variant, or all of the mutations, are located at this receptor binding domain. The Delta variant also demonstrated favorable interactions, and this was noted by the increase in stabilizing hydrogen bonds between both these binding regions of the protein. So next slide, please. So we then looked at the S1, S2 boundary site, and as I said, that is cleaved by furin. So we docked furin to the S1, S2 boundary site of the spike protein, and the docking results indicated that the most favorable binding affinity was seen between furin and the Delta variant protein. And it is interesting to note that the Delta variant protein is characterized by a arginine replacement of proline at amino acid 681, which is seen by the little orange dot that you can see on the far right. And so this alteration, basically the arginine has characteristic positive charges, which then leads to the formation of a salt bridge, which is one of the strongest known intermolecular interactions in chemistry. So this salt bridge is formed between furin and the S1, S2 boundary site, and that the attractive force as well as the increased positive charge has led to the increase in the affinity you're seeing between furin and the Delta variant. So taken together, it's evident that the Delta variant results in increased binding of both the host proteins and suggests a possible mechanism of action of how we're seeing these excessive infectivity rates at the moment. For this study, we only assessed the molecular docking to look at the structural mechanics, but to gain a more comprehensive and reliable data set, we need to look at molecular dynamic simulations. At the moment, we are currently also assessing these variants as well as the newly identified C.1.2 lineage with regard to neutralizing antibody binding and these implications and the implications these alterations may have on vaccine efficacy. So next slide, please. Finally, we'd like to thank all our funders as well as our current collaborators that have worked on the wastewater. Thank you very much. Prof. Angela, over to you.
Thank you, Pratika. Well, firstly to Rabia for helping us just understand how important or powerful wastewater surveillance, in addition to providing an early warning system for COVID-19, to also serve as a system or tool for monitoring variants of concern. And then Pratika for helping us understand why the Delta variant is so infectious relative to earlier variants. We're running a little bit behind time, so I'm going to ask our final speaker of the day, Dr. Carter Farkas, come and share, hello Carter, come and share with us information from the UK on how they're managing the wastewater surveillance system there. Carter, over to you.
Thank you very much. Can you see the screen and hear me as well?
Yes, yes, everything is going well.
Excellent. Thank you very much for the introduction. Yeah, I'm Kata Farkas. I work at Bangor University in Wales, UK. I've been here for 6 years. I'm an environmental virologist. And today I'm going to share you our experience with wastewater analysis for SARS-CoV-2 monitoring in the UK, mainly focusing on Wales. So we do have a background in wastewater monitoring. Over the last decade, probably, we had multiple projects on the detection of aquatic viruses and bacteria originated from domestic wastewater and how they spread and behave in the water environment, and what are the associated health risks like waterborne illnesses. Also in our previous project that I was working on, the Virakwa one, from 2015 to 18, we did a 1-year surveillance of enteric viruses in wastewater and in the environment with monthly sampling, focusing on the Conwy River and estuary in North Wales. And we did find that, first of all, we did find norovirus and cephalovirus signals in wastewater and in the environment. And what was interesting, we also noted concentration spikes in wastewater, which coincided with local gastroenteritis outbreaks, which suggested that monitoring wastewater can be a very effective tool for public health issues and to explore unknown outbreaks. And based on that, we quite early in March last year started a preliminary project, a half-a-year one, focusing on 6 wastewater treatment plants in England and in Wales, with taking grab samples weekly at these treatment plants. Then we concentrated the samples using ultrafiltration, and we used the N1 CDC primer and probe set for the qPCR detection of SARS-CoV-2 RNA, coupled with some sequencing as well. So as you can see, we on North Wales and Liverpool and Manchester. These are cities with quite different populations served by these wastewater treatment plants and with different treatment options. We did use influent samples here and in sometimes effluent as well. And the major findings which have already been published was that the SARS-CoV RNA abundance and diversity does reflect the clinical caseload and the lineages detected in clinical. And we also found that the temporal analysis of the virus in sewage tracked the effectiveness of the lockdown, as the case numbers were quite high before and in the early stages of the lockdowns, and then the RNA concentration went down and then picked up again when these restrictions were lifted. So we concluded that the wastewater-based epidemiology indeed can be a useful tool for pandemic response. But we did find that there are challenges, as noted in the previous talks, and there are other issues to further research in order to have really good data, which is useful for policy making. Following that short project, in September last year, we started a bigger one in Wales, focusing on 19 locations in South Wales and in North Wales, where the majority of the population lives, with 3 to 5 times sampling a week. And we had way more data here and more frequent sampling. So during the second wave of the pandemic in the UK, initiated by the Alpha variant, we were able to note these changes in case numbers using wastewater sampling. We also were able to gain more data and do some normalization for process efficiency, population equivalence, and the dilution of wastewater. And we were able to come up with some predictive models which suggested that we can actually foresee the outbreaks 5 to 14 days ahead, depending on nature of the wastewater treatment plant and all the normalization that we have done. In parallel with the Welsh effort, there are also in other countries in the UK, Scotland, Northern Ireland, and England, there are more efforts being done on sampling sample analysis, modeling, and sequencing. Currently, we are able to, I think, cover 70% of the population overall, with most regions doing at least a couple of days sampling a week or daily sampling. There are different aspects as well, like hospital wastewater or university wastewater, prison wastewater testing as well, to see how smaller communities are affected by the virus. We do have now a preprint on all this if you're interested in what we have been doing all over the UK. As I mentioned, there are a couple of things that we need to consider, and it was highlighted in the previous talks as well. What are the factors that affect the quantification, the detection of the virus, and how that can be implemented in modeling and other aspects. Like it has been mentioned, the grab and composite samples and the differences there. How about the decay of the virus and the viral RNA in the pipes during the time they spend there before they actually reach the wastewater treatment plant where we sample. The sample process, this is the process that we use in the WASH program using polyethylene glycol precipitation. It's laborious, it takes a lot of time, and there are multiple steps where we can actually lose the virus as well. So that's something to keep an eye on and make sure that the sample process that we use is actually adequate for the sample. Also, how you detect the virus. qPCR is the most obvious choice. Many labs do have the equipment and able to perform the assays once they are validated. There is also more and more efforts on digital PCR platforms or LAM-based quantification. More rapid sequencing approaches are used as well. But we just have to keep an eye on these methods as well to make sure that they are properly validated and accurate. Also about changing population, many regions, for instance, where I live in the Northwest, they are tourist destinations and people just come and go, which would then affect the RNA that we detect in the wastewater. And I don't have to mention the evolution of the virus resulting in new mutations and new variants, as it was highlighted in the previous talk. So because we have these challenges, we have a couple of short projects, really focused ones on different objectives to tackle these issues and to make sure that the results that we actually have are accurate and useful. These are on the efficiency of virus recovery or viral RNA recovery, sampling strategies, residence times in pipes, and the detection of new variants and other future applications. So for instance, for the recovery efficiency, my colleague Jess Kevlar has made some excellent lab experience to figure out which method is best to use and what may affect the efficiency of these methods. So here she compared two methods, ammonium sulfate precipitation and InnovaPrep ultrafiltration in wastewater samples with different turbidity and different concentrations of surfactants, which we thought would probably have a great effect on viral recovery. And she did find that the precipitation methods is better than the filtration-based one, resulting in less frequent loss of viral RNA. And also found that the turbidity doesn't really affect the recovery efficiency, but the level of surfactants, if it's really high, it would affect the recovery of the viruses if you use the different methods. And therefore, we highly recommend the use of process controls from the early steps of sample process to really explore that how viruses are recovered, whether they are recovered with the method, or your negatives are truly negatives, or it's just you were unable to detect the virus using that method in that specific sample. The other thing Renee were able to cover, I believe, is the sampling, the graph sampling versus composite sampling, and the use of autosamplers. And actually, it's not only the electricity, it's just autosamplers as they are devices, they are going to break down, usually at the most inconvenient times. They also just sometimes clogged or unable to collect a sample or two for some reason. So it can be really hard to gather the composite sample that would be really useful and representative because it's just really hard to do so. However, graph samples, if not taken at the really good time, they may not be representative either. Now, to overcome these limitations, we did come up with some passive sampler approaches. There have been some use of these previously, not only for COVID, but for other viruses as well. So we decided to use 9 filtration material, which we thought would have the potential to capture the virus in wastewater. We put them in these little plastic devices, which then we adjusted to, we attached them to a string and just throw them in the wastewater and fish them out after either 1 hour or 24 hours in the first instance. And as you can see how the color changes, it clearly shows that over time, the clean filtration material we are going to be saturated with the matter found in wastewater. Hopefully that includes viruses as well. After we saw that this actually can work in a field setting, we also did some laboratory experiments where we could monitor the actual recovery of viruses. So we just collected wastewater and spiked them with SARS-CoV-2 inactivated and the Phe6 bacteriophage, which is an enveloped RNA bacteriophage, very easy to maintain in the lab. And this is our choice of control virus to explore viral recovery efficiency. So we use these two. We also monitored grassphage, which is a fecal indicator virus commonly present in wastewater. So we mixed these spiked wastewater samples with the filtration material, and we did find that there were differences in viral recovery and the attachment of the virus to the different materials. We selected 4 of them, which showed the most potential in terms of viral recovery. In further experiments, which are now ongoing, but also we would need to figure out more about the exposure time. Actually, when these materials are saturated, can we leave them for 24 hours or is it actually too long? And during the evening and night hours, it would just be washed off instead of collecting viruses. And where's the best place to put these as well? Sometimes when you have really high flow, some of the filtration material will be just washed off or you just lose the whole device. So it has to be a safe place, which is representative, but wouldn't result in losing the actual material. So these experiments are currently ongoing and hopefully will give us more information on the correct use or the best use of these very simple devices. Another aspect that we've been investigating is the virus residence time in the pipes. Sometimes viruses can, fecal material, sewage can span days in the pipes before actually reaching the wastewater treatment plant. And during that time, these viruses, especially enveloped viruses like SARS-CoV-2 would eventually degrade and it wouldn't be very representative. So we would just like to figure out how long it really takes for a virus to reach a wastewater treatment plant, which can be done quite easily. However, it is a time-consuming approach. So what we did is that we used the rhodamine dye and the bacteriophage Phe6 inactivated one, so it wouldn't affect the environment. And we just flushed them down the pipes and just a couple of kilometers from the wastewater treatment plant. Then we went to the wastewater treatment plant and collected samples. which were then tested for the dye and for the V6 virus. We actually found that it takes quite long, even if it's just a few kilometers, for the virus to reach the wastewater treatment plant. And with further mapping using experiments like this, we can actually explore residence time better. And then this information can be fed into the normalization and modeling to have a more accurate estimation on wastewater-based epidemiology for COVID. And last but not least, we also developing methods for new variants. Currently, we have in-house QRT-PCR detection methods, duplex assays for the beta, gamma, delta, and kappa variants. These can be mixed and matched actually in using different probe dyes, and they work together quite well. So you can just choose the variant that you're interested in, or the variants up to 3 in one assay, and study them in a very simple 1.5 hour QRT-PCR method, which is a great improvement, as said before, compared to sequencing, which can take weeks. This qPCR can just be done right after extraction, as I said, within 1.5 hours, and you would know if there's a new variant that would be of concern. We did tested this method on over 800 samples that we already had in the freezers, and it seemed to be working quite well, with unsurprisingly the Delta variant being the most abundant variant. We barely detected the kappa, and we sporadically picked up the beta and gamma during the study period from April to July this year. Some of these samples were also sequenced, and we have the sequencing data as well. Now, unfortunately, we couldn't detect the new variants in all the samples that were positive for sequencing, but that can easily be due to the degradation of the sample during storage and freezing thawing, as we just tested archived samples, whereas the sequencing was done on fresh samples. So this is our next project actually ongoing to better understand how long the RNA stays stable during storage at different temperatures, and what are the effects of repeated freeze and thawing, and how long we can actually use the samples that we have for qPCR or for sequencing. So to sum it up, we also concluded that wastewater-based epidemiology can be useful for outbreak tracing and mitigation. The predictive models have extreme potential to project outbreak dynamics. The methods using control viruses should be carefully validated, though, for accurate results. And you should always use control viruses to make sure that everything is going well in the laboratory. Alternative sampling, for instance, passive samplers can be useful when resources are limited once we fully validate their use. And the structure of the sewage system and residence time should also be better understood to make sure that the models are accurate. And also qPCR can be used for the rapid detection of emerging variants, and not only for SARS-CoV-2, but for other viruses and microbes that may pose health risks in the future. And at the end, I would just like to thank all the collaborators we work with within the UK and at the University of Nigeria and RS in South Africa. It's just great, I believe, how we all come together to really tackle this during these very trying times. And I would like to also thank the opportunity for having me here and for your attention for my talk. Thank you.
Thank you very much, Kata, for that fascinating presentation. Especially interested in the work that you're doing on viral recovery and testing various filtration materials and the devices that you are using. And then, of course, also your work on variants of concern and on the transport, the environmental transport in the sewer system. We're running a little bit behind time, but we have just under 15 minutes for any questions. I see there's already a dynamic conversations going on in the chats, and there are some questions in the Q&A. So let's start with that. Faisal Aziz says, thanks very much for the excellent talk, and has he's got a question about the technique, low cost, rapid and precise to detect COVID-2 in wastewater-based epidemiology, and how you can get an idea about the quantity, not only the presence, absence. And I think we've pretty much covered that question. So I'll move on to the questions in the chat. One of the earlier questions was, yeah, there are many, many thanks and commendations for the speakers. Sophie asked, do you know of studies comparing shedding profiles of different SARS-CoV-2 variants? Rabia, will you be able to take that question for us?
Yes, Angie, just where's the question? Just repeat it.
It's on the chat, and the question, Sophie's question is, do you know of studies comparing shedding profiles of different SARS-CoV-2 variants?
There's lots of data currently in the public domain that you can have a look on, specifically with pertaining to wastewater-based epidemiology. Clinical data, there is lots of data for clinical data, but for wastewater-based epidemiology in South Africa, there's no current data for the variants of concern, except for the genome sequencing data that is out. But internationally, yes, there are definitely that you can do a comparison between the different wastewater treatment plants, and like our data showed, we are able to compare the different wastewater treatment plants. So what I've presented here is data from 17 wastewater treatment plants, and we can look at the wastewater treatment plants to compare what's the variance that's currently circulating within those wastewater treatment plants. Yeah.
Rabia, please stay on the line, because I think you're also the right person to answer the next question from Professor Himla Sudel. And she's saying, from a pooled sample, how do you get individual sequences? Or are you sequencing from pooled samples?
Okay, so remember the when we are sequencing from wastewater, that's not a pool sample. It's in a sense, it is a pool sample because it's nucleic fragments from various individuals that are shed. So what we are seeing, we give an indication, a holistic view of what's happening within that catchment area, of what's happening within that population. Whereas if you sequence a clinical sample, it will be one clinical sample is equal to one genome that's being sequenced. Whereas if you take a wastewater sample, the wastewater sample will give you a holistic overview of what's happening in that population, what strain or what variant is currently circulating within that population. So it won't give you an indication of a individual, but rather of what's happening and what is currently being shed within that wastewater treatment plant. Yeah.
Thanks very much, Rabia. Then Renee, there's a question from Dapo at DG. Do you use absolute quantitative? I think, have you seen the question, Renee?
Yes, it's been answered. It's been answered by Rabia on the chat. Thanks.
Okay, all right. Thanks very much. Then Kim Nguyen's asking Carter a question. Can you share your insights on the role of wastewater surveillance, taking into consideration the presence of herd immunity?
Sorry, could you repeat that? I didn't get it.
I think I'm not quite sure. Please, could you share your insights on the role of wastewater surveillance, taking into consideration herd immunity? I think that's the key question.
I think it's how the vaccination affects basically wastewater-based epidemiology. This is interesting because the preliminary findings that we have, this has not been published yet, was that in many places, even though the case numbers are low, which was probably due to the vaccination efforts, we still had quite high numbers of SARS-CoV RNA in the wastewater. I think one of the talks also mentioned that maybe Renee's talk. So that's something to consider and further explore why that is. It can be that because the Delta variant is just so much more infectious and it's also shedded in higher concentrations. So actually the case numbers are not as high, but we just see more virus coming from one person in wastewater, or it can be that there are more symptomatic infections because people also have some immune response because of the vaccination. So I think that's something that needs to be further investigated globally, really, and not only in wastewater surveillance, but in clinical settings. I'm sure they are investigating how that goes and what's the actual rate of symptomatic infections when you're vaccinated or had a previous infection.
That's right. That's right. I think time is going to answer that question much more effectively than we can right now. Renee, can you respond to the next question? Anatolia is wondering about the implications of the presence of the virus in wastewater on personnel working in the plants and also on reuse of the treated wastewater. Perhaps you can just chat about our vaccination program for our field staff in South Africa as well.
Yes, sure. So what we're finding in the wastewater is only the viral fragments of SARS-CoV-2. It's non-infectious and it's the inactivated virus. So there's no concern about any of the sampling staff or the wastewater treatment plant staff getting COVID-19 from the wastewater. However, they do need standard vaccinations to be handling wastewater, such as hepatitis vaccinations and tetanus. So we do make sure all of our staff on the program are vaccinated for that, but it's not a concern to get infected by COVID-19.
Thanks, Renee. Himla has a question about why we're not routinely using saliva samples for virus detection in individuals instead of the nose-throat swab. I'm not sure if anyone on the panel would like to answer that question, or if it's already been.
I can try, Angie. So there are papers out on where they are currently using saliva, but those papers are showing that that there's a decreased sensitivity with the saliva. The nasopharyngeal swab sample still remains the method of choice and the best method. But I also know that there are currently labs that are mouth swabs, cheek swab samples, but then it is not as sensitive as the nasal one, even with the saliva, because it depends on how much you're getting. Are you just getting sputum? And that's why when they did sensitivity and specificity, some of these papers found that there was a decreased sensitivity and specificity using just saliva.
But yet we can detect it in wastewater.
Yes, that is what the paper said. We haven't done it with saliva.
I heard a talk given by somebody who was partly responsible for developing the technique at Yale University, and the efficacy there was more than 99%.
Yes.
So I'm wondering, why you feel that it's not as sensitive.
There are some papers, so there's contradictory information out there if you read the papers currently. So some papers would say it is sensitive enough, whereas others would show that it's not as sensitive. It brings me back to TB. With TB, for example, I'm just making an example of another disease state where you use saliva, and depending on the type of where you get your sample, whether or not you're able to extract the bacterium from the saliva. So yes, you can detect it. I'm not saying no, you can't. It's just some papers, there's contradictory information out there, but it would be a better option than the no swab sample. Yeah, I completely agree.
Thank you.
Thank you, Rabia and Himla. So as we're moving now to closure, I would just like to thank again to all the speakers. It's really been a fascinating webinar covering the challenges experienced in all three of the countries, the extraordinary challenges experienced in Nigeria. I mean, to be able to have a wastewater surveillance system, you actually do need wastewater treatment plants, and it is disheartening to see how few well-functioning wastewater treatment plants there are currently in Nigeria, and it just brings to the fore also the importance of environmental sampling. So good to see that Vincent intends working on that issue. That can help many, many countries, and also the work that Kata is doing on the same issue in the UK. All of the presentations together just reminds us again so powerfully how important wastewater surveillance is in public health. And it's a tool or a framework or system that we have neglected, I think, for decades. And we've just seen today how the wastewater system is useful, how useful it is as an early warning system, also as a device to monitor changes and patterns in the different variants. And we haven't had a chance to talk about it, but it's possibly a topic for another webinar, how wastewater surveillance can extend beyond SARS-CoV-2 to other systems, to other issues or other issues of concern, especially in Africa, around chronic diseases and pollutants, etc. It's been wonderful to see how the scientists on the webinar today have responded to some of the challenges around the absence of information, on shifting from composite samples, which is the gold standard, and doing the research to show that in most contexts, grab samples are highly robust and can form the basis for an important and powerful wastewater surveillance system. And so we look forward, Himla, I see your hand. We look forward to the additional future research coming out of this team and scientists like we have on the webinar today, and to inform or help us develop wastewater surveillance systems beyond SARS-CoV-2. Himla, would you like to come in now?
Yes, sorry, Angela. Given that Prof. Musa Moshabela had joined us just after I had spoken, perhaps if you could just give him 2 minutes to say a few words at the end. Thank you.
Absolutely. Prof. Moshabela, please go ahead right now.
Thank you so much, Angela and Himla. I want to take this opportunity on behalf of the Standing Committee on Health at ASSAf to thank you and the team for putting forward this submission for this webinar. It was fantastic and it was so important. It is so timely. And I think that, as you correctly said, it's a tool in our toolbox of epidemiology that we had left out and neglected. And we really need to think about how to bring it to the front once more. And I was so impressed to see that this program has grown this much at the MRC. And I really hope that we can continue to highlight it. And I can see how when we bring the different surveillance systems together, the story becomes complete. It's almost like, you know, the investigations that we are doing are able to add another layer of information and knowledge that we can then be able to take forward and advance. It's really interesting to see that you can actually go back to detect where these variants were seeded, for example. And that story is something that has been difficult for colleagues who are doing genomic surveillance to be able to tell. So I just think that, you know, this is so fascinating. I'm really looking forward to hearing more about this work, and you know, also making it a lot more widely available publicly for people to become aware of it. And wish you all the best in the work that you're continuing to do. I saw some colleagues were saying that you need more funds to take it forward, and I wish you luck in getting the funds necessary to continue the work. But once again, on behalf of ASSAf and on behalf of the Committee on Health, thank you very much to you and the colleagues, and to all the attendees. Much appreciated.
Thanks very much, Professor Moshabela, for those words. And we couldn't agree with you more that wastewater is a treasure trove of information for public health. And we really do need to do a lot more data mining around wastewater surveillance. There have been several requests for recordings of today's webinar and for the presentations, and I think Kutso has responded on that score. So finally, I'd like to thank all of the participants today. Thank you for joining us to hear about our research and our experiences from these 3 countries, South Africa, Nigeria, and the UK. And finally, again, thanks ever so much to all our speakers. Thanks, everybody, and goodbye.